Okay, so in order to sample the shoe in the protocol that we happen to be using, you need your shoes that you want to sample. You're going to need a collection tube that has some of the preservative in it, and then you're going to need a sterile swab in order to do the sampling. You don't really need gloves. I mean, you might want to protect yourself from the things that are on the shoe, of course. But as long as you really make sure that you keep the swab anywhere that you keep it out of anywhere that you don't want it to be, you can just go ahead and do the collection at a tabletop. Students can do it in the classroom and in the lab, or they can take it home and do it. So it's fairly simple, but I'll demonstrate it and also just point out a couple things. So we have the collection vial. I'm going to go ahead and label it. Okay, and if we're following good practice, since I've given myself a number, I'm going to try to label the top of the tube as well. It's a little harder to write on, but I can just make out numbers because caps and tubes can become separated, so it's good that everything is labeled. And then for the shoe itself, I don't know that there's any fancy trick. I have a left and a right shoe. I don't know that either will yield different results, but that's what I have. The main thing that I find, one, is if you were to take this swab and just rub it against the shoe, you get something. But what I actually think I find is that wetting the swab in the solution first, and then doing that wiping yields a better result. So what I'm going to do is carefully manipulate the opening here, get it out. Okay, and I will take my cap, put it off to the side, and I'm going to go ahead and dip it in. Okay, I'm going to recap it, kind of thinking about sterility, but this is not a biosafety level 2, or this is not a forensics lab. So I do know that I am, there may be places where I can optimize, but like we said, this is for education. Now, I find that it's nice to go all around the shoe. You may notice spots or clumps of soil or whatever. These are gym shoes, right? So technically, I don't think I get so much exposure to, let's say, dirt. But maybe they'll look pretty different than other shoes. I don't typically go inside of the shoe. Again, you probably would expect that surfaces that are frequently in proximity to us as humans is also going to have microbes that are associated with us. But you get the idea. What I do try to look for is that the swab has a visible color. Might be easier to see a little bit against this background that it's now looks dirty, right? And so that is the first clue that I'm going to get what I'm looking for. And the interesting thing here is that if you take this swab, you can put it in. Again, we don't want to touch any other surfaces, just the inside of the tube. When you put it in, you can kind of almost wash it off in that solution because we're going for the whole thing. But the other thing to notice, it's a little bit hard even for the zoom in, maybe you can see it there, is that there's a little break point on that swab. And if you put this in, you can snap it such that you can pull out half the swab and cap it. If you cap it just right, the swab actually fits in a little hole that's in there, and then it stays upright. I mean, not that it would completely sink, but this is what it looks like in that tube. Can show it there for the camera as well. But that is our sample.